And it's official, we're being recorded. All right, thank you very much. I'm Jinsy Andrews, and I am really happy to present today and really honored to get the opportunity to do so. Of course, there are many challenges in drug development for all diseases, but there are special ones for ALS, and I, I am going to try to share my experience. So some of you may already know, some of you don't know. I trained at Columbia as a neuromuscular fellow and and did my clinical neurophysiology fellowship as well, and then stayed there. I was recruited out to the University of Connecticut to start a clinical trials unit from scratch. So that means building a contracts, finance, and a clinical trial infrastructure. And so I learned a lot about administrating over clinical trials for all neuromuscular disorders. And then from there, I was recruited to a biotech company, Cytokinetics, where I really did get a little inside view on how pharmaceutical companies view drug development and what's valued when you're trying to bring something into a industry and develop it into human clinical trials and commercialize it. And then I was recruited back to Columbia to oversee clinical trials in ALS in 2016. So I'll try to share some of that perspective today. These are my disclosures. I served as a DSMB chair this year for ALS Pharma. I've served as a consultant for many of these companies. Only when I hit any of the investigational products during the presentation, I'll call out that if I have served as a consultant, and I've received research funding from several sponsors and foundations. Today, I think in challenges, I'll try to focus on three main areas that have been really tough for ALS in particular. One is choosing an ideal drug target. I'm not a basic scientist. I'm a clinical researcher, clinician, neurologist. And so there are a lot of you that are out there that are specialized in the targets and the biology of whichever areas that you're focused in. But there are some concepts when you're looking and evaluating those specific targets that would make them ideal for developing them further, moving them across into clinical development or partnering them with pharmaceutical companies. I'll just highlight some challenges in developing outcome measures and not necessarily talk about outcomes in ALS because that's another talk in and of itself. But when you come to regulatory review, what are they looking for when you're developing outcome measures? And then the last, I'll provide the evolving regulatory landscape in ALS and how it's really shifting as we speak today. So what's an ideal drug target? And this is a complicated topic, and I'll probably spend a little bit of time here. A lot of it's a high level conceptual review. So the hurdle, really only 4% of drug development programs yield a licensed product. And in ALS, for all of us who work in that area, know that it's a struggle. Preclinical experiments and early phase clinical testing to support target identification and validation are really poorly predictive of eventual therapies that are successful. And we struggle sometimes to find definitive evidence of validity. And that's because of many, many factors. I mean, the core of it is that our understanding of the biology of the disease still needs to be added to for ALS. Sometimes when we're doing preclinical testing, we have to be mindful of how we're blinding those experiments and how we're randomizing them. The better the rigor in the preclinical experiments, the better the predictive value in human clinical trials. There are some hurdles that are just plain challenges, like species differences, inaccuracies of animal models to human disease. And then statistics. Statistics is really important in preclinical testing and also in human clinical trials, and I'll try to highlight on both levels, but over-interpreting nominal significant experimental results will sometimes carry this false logic of carrying a target forward and then eventually going into clinical trials and failing. Let's see here. When we go to human observational studies to help us with validation, what happens is there's a lot of confounding variables. I mean, for one type of example in ALS is looking at lipids and trying to figure out whether lipid levels, lipidomics, and how diet and exercise and genetics all play a role can be really difficult to tease out. And the other major issue in logic in terms of carrying targets forward is reverse causation. Now, reverse causation, it's like when a risk factor causes disease. We know that the risk factor is associated to disease, but that does not necessarily mean that it's a causative of disease. We might know that a risk factor is correlated with disease, but that does not infer that that risk factor causes disease. Reverse causation, for example, say in patients who are depressed, you see that they tend to be smokers. So we think that the risk factor for depression is smoking, but maybe depression itself might actually cause people to smoke more. So there is a very careful descriptions about how to establish causation in epidemiology. And often in ALS, it's really difficult to establish causation of risk factors. Evidence of target validity from phase one clinical studies can also be inadequate. I mean, phase one, we're really looking for safety and tolerability of a compound. So often these studies are very short in duration, they're very small in size, and are often, it's often difficult to kind of validate targets in that early phase. So sometimes you're moving forward to a larger phase study with some uncertainty and some risk, and it costs money. And if you advance something that might not be true, you can lead to a late stage clinical trial failure. Now, all of you are very well versed in this, but target identification strategies, even as I worked in my industry position, the most common strategies that were used were looking at literature review. Of course, scientists all over the world are working on different pathophysiological mechanisms, looking at focused proteomics, pathway analysis, and only when I entered industry did I understand that there were many databases available to look at pathways and phenomic databases, doing functional screening and genetic associations. I think the hardest part in ALS is from a pharmaceutical perspective, the identification strategies that are successful come from really understanding the disease and the access to really good models and technology and the clinical data, natural history data that really helps to identify a promising drug target. But as you all know, in ALS, we struggle to fully understand all of the aspects of the disease itself and the biology. We're still working on better models and technology, and we're trying to improve on that so that when we carry something into clinical trials, we can be more successful. In validation, from validating a target, you really have to consider biomarkers. Our ALS community as a whole have been focused on trying to develop targets and then trying to find meaningful biomarkers that can that can confirm the target engagement. So here's a couple of examples. We have actually a growing biomarker. You have a biomarker talk coming, so I don't have to lay out all the biomarkers in ALS, but just talking high level, if a mechanistic biomarker suggests that a target was insufficiently hit, then that would, in the lab setting, would make you want to look for a better lead structure. That's pretty simple. But if the biomarker confirms sufficient modulation of a target or a pathway, right, but you don't have a net clinical benefit in that disease population, then you got to ask about the validity of the pathway. So let's say, for example, in hypertension, this was a good example of where the angiotensin receptor antagonist in healthy volunteers in phase one didn't lower blood pressure, but it did increase the plasma renin levels. So it showed that the target was engaged, the pathway was engaged and relevant in the human setting. And that led to successful clinical trials. But say for ALS, we have a lot of biomarkers that we look at in the spinal fluid that we're evaluating. If you look at that biomarker, and it's something specific to the drug that you're using, and it hits it, and you're able to increase it and it decreases over time as you repeatedly give the drug, you have to think about whether that mechanism is valid. And if it is, because you're giving the drug repeatedly, but it doesn't sustain the increase in the biomarker that you're looking for or the decrease, it could be that the human system itself is regulating it in some way, or even more complex is if you're moving these biomarkers, but you don't have a net clinical benefit, you have to reevaluate what that biomarker means. Because I don't think in our ALS community, we look at a lot of biomarkers, we get very excited, but we have to fully understand, are they predictive biomarkers? Are they prognostic? Are they very specific to a mechanism for that specific drug? but does it have a value over the survival or function of ALS patients? Those are questions that need to be answered to fully understand what that biomarker is. So I think we're a little loose in our vocabulary, and I think Mike Benatar will talk a lot about that. So when you're looking at ideal drug targets, from a pharmaceutical perspective, there are three real key concepts when you're doing an evaluation. So when pharmaceutical companies come to an academic lab and look at it, they look at three key features. So it's the druggability of the target. So a druggable target, this is actually a Steve Finkbeiner. He actually talks about it too when I went to the California ALS Summit. A druggable target is a protein, peptide, or nucleic acid with activity that can be modulated by a drug. So that can be a small molecule or a biologic. And you really want to know your chances of success are better if you understand the structure of your drug target, and if you have if you can see if there's any predictability if you're working with a small molecule. So my background is I worked with small molecules. The predictability is better if you can find the structures and find potential binding sites for the small molecule. Assayability is a very key component. So this is a biochemical and or cellular assay for binding or function to support screening program suitable for your lead compound. If you don't have an assay, it's very difficult to kind of move it forward into human clinical trials. And then another topic that usually doesn't come to mind readily when you're working and trying to validate targets and look at targets in an academic setting is the freedom to operate. This is intellectual property. And so when industry evaluates a potential target, they'll look to see if that particular action or that assay can be done without infringing on other valid intellectual property of others. How competitive is the market? How big is the unmet need? How many drugs are there that are similar to that mechanism? Is it first in class? What's the risk? And what is the potential to operate without infringing on other IP? So you can get intellectual property on the compound or intellectual property on the use of a compound in a specific disease. So there are, and of course, pharmaceutical companies look at the assays and see if those assays can also be owned as intellectual property. And those are really favorable factors for companies coming in and trying to partner with an academic lab on a particular target. Now, this is a good example of an evaluation checklist when you're looking at a potential target to develop in a clinical development program. So if the target is something that's in a disease with high unmet need like ALS, it's very attractive. And then, of course, you identify the molecular target, and then the target assessment, you want to look in the experiments to see if the molecular mechanisms have been characterized and addressed by the target. And are you able to modulate that target in a relevant in vivo model, like a knockout or transgenic mice? But there are some theoretical assessments that are done for the target. So that's the druggability concept. Does the small molecule binding domain exist? That bodes better. Is there an extracellular domain for the biologic that you're evaluating? Is there a crystal structure available, and is high throughput screening available? These are very attractive when you look at druggability assessments for potential targets. And then the idea on target related or stratification biomarkers will help to either ensure early proof of concept or help stratify your human clinical trials. Another real major component of evaluating a potential target is looking at the adverse event potential. So when you're looking at that target, how selective is the tissue that the target is being aimed for? So if the target aims at tissue all over the body, then there's a high likelihood of off-target effects or adverse events. Phenotype data, clinical data, and sometimes if you're developing a target and investigational compounds that are within a class, so there's other drugs that are already out there that have been put into humans or have had animal experiments, you can compare its adverse event profile to the similar class compounds. Now, freedom to operate, intellectual property is a very important concept, like I mentioned. So things like freedom to operate analysis occur with the business teams on the pharmaceutical company side. The options for commercialization. So we talk about ALS, the disease has a high unmet need, so it's a good opportunity for commercialization. But if your drug targets a very narrow population or has high cost of good manufacturing costs, it might not be feasible to carry it out in a clinical development program. Is there a common mechanism potential for the target? So meaning the target that you're looking for, it's good for ALS, is it good for other diseases? That makes it very attractive as well. And then again, looking for options from a business side of generating intellectual property. So whether it's the chemical compound itself and the structure, the use, or the assays, those are very attractive. And so this is just a compiled list of all of the things that kind of compose an ideal drug target. So obviously the target, it's ideal if it's disease modifying or has proven function in the pathophysiology of the disease. You can modulate that target, and modulating that target is less important under physiological conditions or in other diseases. So meaning it's specific for the biology that's making the individual with that disease sick. Is if the drug ability is not obvious, a 3D structure for the target protein or a close homologue should be available for drug ability assessment. Again, assays are important for high throughput screening. If a target is not expressed uniformly throughout the body, then that's better and more specific. A biomarker, that's like the holy grail for ALS. If we have a biomarker or a disease-specific biomarker, that's always helpful in trying to establish proof of concept. A favorable prediction of potential side effects according to the phenotype data, and then a favorable intellectual property situation. So essentially, the big picture here is discovering and assessing the potential therapeutic benefit of a drug target is based not only on the experiments and the mechanistic and pharmacological studies, but you also need to think about the theoretical drug ability assessment and early assessment of potential side effects and any opportunities for commercialization, the intellectual property component. And then I just put this up here because all that concept from the pharmaceutical side, we've been able to kind of harness into Columbia. This is just an example, and I'm sure you guys are in the ALS clinical trials unit at Johns Hopkins. It's very similar. Project ALS has allowed us to kind of bring together our specialties within Columbia and the Motor Neuron Center and really crosstalk to try to do all of the assessments that we talked about just previously. So we do pharmacokinetic, pharmacodynamic measures, we do medicinal chemistry, and we have go, no-go criteria to move things forward. And I'm in charge of the clinical unit so that once either trying to bring samples from patients into the lab to try to see if there's drugability, screening, but once a compound goes through its necessary testings, I can translate it into clinical trials. And so it's led by Serge Preborsky and Neil Schneider and Hinek Wichterly, and then we have core unit directors. And then the director of operations, Emily Lowry, helps to kind of connect us all and keep the crosstalk going regularly. This has proved over the last 2 years of its development to be pretty efficient and pretty productive. And so we do have the FUS antisense oligonucleotide program that's now being developed for clinical trials with a collaboration through Ionis and Neil Schneider. Procetin, which is a MAP4 kinase inhibitor. There's a publication in Cell Biology. It was screened and optimized at the labs at Columbia, and that is now undergoing IND enabling study and really is an example of how the core brought all of those components together. And then also collaborating with sponsor Cellenkos for potential allogeneic Treg therapy for first-in-human trials in ALS. So with that, you find a good target, you look through its drug ability, its assay ability, you realize that it has good commercial opportunity. So you're trying to bring it into human clinical trials. How do you do that? And so I bring up the idea of clinical endpoints because sometimes you do functional assessments in animals and it's hard to translate into ALS. I think this is a major challenge for us. Survival is our gold standard in both animals models and also in human clinical trials, but doing an ALS clinical trial with survival as an endpoint is difficult because it has to be lengthy and it needs large sample size. So I just wanted to take one example that I've done at my industry position. So this was an animal model. So I worked on a small molecule trying to enhance skeletal muscle function and strength in the setting of diminished nerve input. And so this is a rat EDL that was stimulated, and we measured the force that was generated pre and post dose for the skeletal muscle activator. This is for tirasemtiv, and we did it for reldesemtiv. And so we were able to stimulate at repeat stimulation frequency and see that post-dose, we were able to increase the contraction force of the EDL of the rat. Now, we had to figure out how to replicate this pharmacodynamic finding in humans. And so what we did is we were able to create a device to measure force. So if you stimulate the deep fibular nerve, you get a contraction of the foot up, dorsiflexion. So the foot goes up, and when the foot goes up involuntarily because the nerve is being stimulated, you can measure the force that's generated. And so we were able to kind of replicate that animal experiment. And what we did is we went to a clinical trials unit specifically for phase 1 clinical trials, recruited healthy male volunteers, and we were able to see very similar pre and post dose. This is concentration based findings, and it's published in Muscle and Nerve. But I flew to Indiana into the clinical trials unit and stimulated the nerves, and we measured all of these, and were able to kind of replicate the pharmacodynamic measure. Now the trouble is, this skeletal muscle activator is not specific for ALS, so ALS biology, but we wanted to take it into ALS. The trick here is that now we want to do a trial in ALS, and doing this experiment and this particular measure is very difficult for ALS patients. And so it was hard to translate that pharmacodynamic finding for ALS patients. Now, this is just a list of typical endpoints that are traditionally evaluated in clinical trials. And so there's a functional rating scale, there's a breathing measure, and breathing is important because that's what people die of in ALS. And we do muscle strength testing, quantitative muscle strength testing, because muscle weakness is the burden of disease for ALS. And a lot of these have been developed and used to assess function in patients living with ALS, and really to get away from using survival. Riluzole, it was the first FDA-approved medication for ALS, but that was based on survival, and the studies were 18 months long, and patients were randomized to placebo for 18 months. And so after that, there was really a push in the community to find something better. And so this functional rating scale was done to help come to an answer about efficacy faster, but also to reduce sample size and hopefully reduce exposure of patients on placebo arm. Now, this is just an example. Here, if you do, this is statistics, but it's just an assumption of a two-arm treatment versus placebo, 1-to-1 randomization, 5 visits, say over 9 months. And we make assumptions of mortality or loss to follow-up, and you have 80% power to show a 30% slowing of any of the outcome measures I just showed you. This is the amount of patients we need. And this is just an exercise to say that we could probably still do better. And so there's been pushes, and I'll just use a couple of examples of outcomes that newer outcomes that we're trying to do to see if we can use less number of patients or less observation period to see if we can find an effect or a signal, a preliminary signal of a pharmacodynamic effect on ALS. And so Stuart Rutkov has worked on electrical impedance myography. This is a really nice technique because it's painless, it's non-invasive, and you can apply it to any superficial muscle. And it's sensitive to changes in the muscle composition and structure and atrophy, and you get voltage that's measured. And essentially, looking at this compared to other things that we typically use, like ALS functional rating scale and HHD, you can see in the top left-hand corner, the slope, you see a decline of the steepest slope. So the steepest slope is where you find the muscle with the fastest change. It occurs much more rapidly than some of our more traditional functional measures. And so the hope is that we could use that as a potential signal for like phase 2 or early phase clinical trials. The coefficient of variation is very important because the larger the variability of that measure, the more number of patients you need to find an effect on that measure. And so that EIM, electrical impedance myography steepest slope, had a pretty tight coefficient of variation. And ALS, FRS, and HHD are not bad measures per se. But this is just an example. If you do the calculations, assuming a 6-month study that's placebo controlled, and you do a 20% treatment effect, which is typical, you can see that the sample sizes needed would be far less if you use the EIM steepest slope. Now, the issue is we get really excited on novel outcome measures, and even with things like quantitative muscle testing that we routinely use as secondary measures or breathing measures, there's a lot of questions from regulators when you take it for regulatory review. So if you see a 20% difference in even the ALS FRS, or if you see it in the muscle testing or breathing testing, what does that mean? And is it clinically meaningful? These are difficult questions, and there's a very rigorous definitions from the regulatory perspective, both in the US and in Europe. And so is 20% difference, 30% difference? What is the minimum difference that you would need? And what is its relationship to function and survival? So while I was developing a skeletal muscle activator, we had impact on the vital capacity. But at that time, vital capacity, especially slow vital capacity, had very little published on its relationship to survival and function. And so regulators certainly did not see it as a surrogate marker for function or survival, and required more data to be published for it to be a relevant endpoint for approvability. Um, and the other thing that I think is growing in importance is the patient perspective. So how much of a difference in any of these endpoints are enough for a patient to perceive an effect in the way they feel, the way they function, or the way they survive? Um, and so another variable that is being explored by Dr. Scheffner in oral analytics is the speech variables. So I think in ALS, we look a lot on physical function based on fine and gross motor, but when speaking and swallowing functions are affected, it impacts quality of life, but we don't really have great measures to do that. And so using technology that can be used at home, actually, you can look at articulation or syllables over a length of time, and there's been good correlation. Remember, I talked about the strengths and weaknesses of correlation, but there's good correlation with the speech, ALS-FRS speech scores, and change over time. Another example of looking at bulbar strength is the Iowa Oral Performance Instrument. So this is like a little bulb that goes in the mouth, and a patient has to use their tongue to press it up, and it generates a pressure. And this is a very easy machine to use as well, and is being used in clinical trials. But we have a lot of questions still, because these measures, even for the bulbar assessments, we don't really understand what changes in these measures are the meaningful from the patient perspective. How much speech loss is a cutoff point for them to be correlated with those changes in pressure by the IOP or the speech generating analytics. And then these changes over time, we still have to understand, and they're being incorporated in clinical trials. And the most important thing is, as we're developing these measures in the community for outcomes, for function, ideally, from my perspective, what would be great for ALS is for clinicians to talk to preclinical scientists and people doing animal studies to see what are measures that we could do across both, like electrophysiological measures. Can we do them in animal models and in humans? Functional measures, can there be functional measures done in rats or mice? And that can be done also in humans in a different way in the ALS population. Those would be ideal. And then having those correlated or associated with function and survival would be the most ideal scenario when you're looking at outcome measures for easier road to approval. So these are things that I think the community as a whole, and outcomes can be a whole talk in and of itself for ALS. But this all kind of comes down to the regulatory landscape for ALS. So from target identification all the way to human clinical trials, the regulatory landscape influences what you would need to do to get it to move forward. And so I think it's important right now to have these discussions with all people working in ALS. As all of you may know, ALS had a drug development guidance that was finalized in September of 2019. And one of those things is the risk-benefit considerations. And this is a quote directly from that guidance, which states that when making regulatory decisions about drugs to treat ALS, FDA will consider patient tolerance for risk and the serious and life-threatening nature of the condition. This implies that there could be some regulatory flexibility for the effect size, the tolerability and safety profile in the context of ALS being a life-threatening and serious condition. Now, there have been recent approvals outside of ALS that demonstrate that there's a transitioning and innovative kind of regulatory framework, especially for life-threatening and serious diseases. And there's two main concepts that are driving it: speed and innovation. And as you know, of course, we're all experiencing and living the COVID pandemic, and we see that the FDA has had to work in an expeditious way to get vaccines approved. But the approvals really for things outside of ALS that represent the transitioning regulatory framework really reflect the patient voice and patient-centered drug development, the data quality, and the evidence that's generated. So in that regulatory framework, FDA has taken things like the use of expedited pathways with post-approval data. They've allowed for innovative trial designs, including adaptive pathways and endpoints, endpoint modification driven by patient-focused drug development, and use of real-world evidence. Real-world evidence means drug is approved. Afterwards, you collect data as it's being tried in the general population of that disease, and that can add as supportive evidence for the drug approval. And so one such approval is Exondys 51 for Duchenne muscular dystrophy. Now, this was approved based on changes in dystrophin, but there was some question about the approvability of that particular endpoint. And so it did go to advisory, and the power of the patient voice, the relevance of the disease and how it's affected, the unmet need, all of this was taken into consideration in its approval. There's a lot you can read about it, but there were three main points that were used in this approval pathway that flexed the regulatory decision. And so one is the use of fast track designation. Fast track designation, usually you apply for it as you're developing your investigational product, and it facilitates the development and expedites the review of the drugs that are intended to treat serious conditions, demonstrate the potential to address an unmet need. It also, this FDA approval in this specific example used priority review. This priority review is granted to drugs that, if it's approved, will be of significant improvement in safety or effectiveness in the treatment of a serious condition. And then orphan drug designation, it provides incentives such as clinical trial, tax credits, PDUFA user fee waivers, and marketing exclusivity. This is really important when it comes to industry developing a compound, especially in ALS. ALS is considered an orphan disease. If you have a target and you validated that target and you have an investigational compound that interacts or modulates that target, one of the earliest things you can consider is an orphan drug designation and applying for it, because this does make that particular investigational small molecular biologic attractive to pharmaceutical companies for partnering because of the market benefits. And again, this was approved under an accelerated approval pathway. So there's the full approval pathway in FDA in the US, and then there's the accelerated approval pathway. In Europe, there's the conditional approval pathway. Those are the only two pathways that currently exist for drugs to be approved and commercialized in the US. In the traditional paradigm, typically, if you have a study that met the 0.05 statistical significance on the primary endpoint, you'd have to do a second study to confirm the first study. And with the two positive phase 3 studies, you would then apply for FDA review and approval. In the accelerated approval pathway, you need high statistical significance on either the primary endpoint, which is approvable, so it has to be stronger than 0.005, or you have to have a strong statistically significant effect on the treated arm versus a placebo arm on a surrogate marker. And that surrogate marker has to be credible and validated through the FDA. So that means that if you're using in ALS, if you're not using survival or ALS FRSR as your endpoint, whatever endpoint you're using has to have an established relationship to survival. And that meets the FDA's criteria. And so we don't have that yet. And they highlight that in the guidance But that's one way where the community can help to improve the regulatory pathway to be more efficient for ALS drugs. The second example, and this is an example I put here because it relates to what's happening in clinical trials with ALS. So Keytruda is a treatment for cancer, and it's a programmed death receptor blocking antibody. But it received its approval because it is a common biomarker, PD-1, for several cancers. And so it was based on this trial where the biomarker, rather than the location of the tumor and the tumor size, was the endpoint. So there were different types of tumors that had a common biomarker. They call it a basket trial. And it showed an effect on that biomarker with multiple diseases with a common biomarker, and it received FDA approval. But it was really risky for the company to develop. So the benefit risk was balanced, and that was evaluated and discussed with by the FDA because different tumors from different areas carry different side effects, adverse events, and safety profiles. Decisions regarding investment and commercial viability by the sponsor. So you're taking that risk. Is it viable to look at, and is it possible to look at different disease populations and with a common biomarker? Is there clinical trial networks in those different disease and tumor categories? And this is relevant to say ALS, for example. I'll just say a basket trial can be applied to C9orf72, where you have a population of patients that may just have frontotemporal dementia. You may have patients that just have ALS, motor weakness without dementia. And you can look at a common marker or a common marker for both diseases, which could be poly-GP or DNP. And so that could be a way a basket trial could be conducted for ALS. When you do these things, especially with biomarkers, there is no regulatory predictability. So it really does take establishing the literature. It's critically important, actually. If you're looking at any targets and looking at any markers, there has to be published literature about it. And it doesn't have to be that strong, but it has to be out there that there's a relationship, because that's what they look for in evaluating the credibility of biomarkers.
Jinsy?
Yeah.
Is Jeff. Using the example of C9, though, wouldn't you first have to prove in a clinical trial that GP is associated with clinical efficacy? Then when you have that, you could then say, great, then it applies to, say, FTD.
That's a great point. And I think that's where, so I made the jump already, and that's kind of what I think we do sometimes with ALS, but that's a very good point, Jeff. When we're developing those biomarkers, we have to establish those relationships with those specific disease entities, and then you can tie them up into, say, we can call them a common biomarker, and then do the clinical trial. You're right.
Yeah. Oh, no, I say that only because GP is a good example. I know everyone's been trying to use neurofilament. Right now, that's not been working.
Yeah.
GP looks to be one of the best ones, assuming it'll track with a clinical efficacy.
I hope so.
Yeah, I do too.
Yeah, that's a great point. But this is where I think we should leverage these things, because it's possible for us to move quickly in multiple disease entities, if possible, if there's a crossover. And so I talked about basket. That's a single investigational drug for different populations. There's umbrella trials where you have multiple investigational drugs for a single disease population. That can be done either as a single drug tested, or you can test combination drugs. For ALS, we've chosen to move with the platform trial design, and there's actually publications in the New England Journal from Janet Woodcock actually about these different trials and how these can help with efficiency and speed of finding the true efficacy of a drug. So in platform trials, there's multiple investigational drugs. So these are single drugs from different companies entered into one infrastructure for a single disease population, such as ALS. And so this is just a quick snapshot. This platform trial discussion and clinical trial could be a whole talk in and of itself. But I just wanted to put a snapshot here so you can see the concept, which is essentially entering multiple drugs in and essentially sharing the placebo arm. So the idea for the platform trials in ALS is a 6-month placebo-controlled study where there's a 3-to-1 randomization, so less chance of exposure for patients to be on placebo, more chance to be on active drug. But when you do the analysis, you can pool the placebo so that you can kind of save on exposure to placebo. And then after that, patient can complete after 6 months, either re-enroll if they're still eligible by the inclusion-exclusion criteria, or they can proceed to the open label extension where they get access to the investigational drug. And I just put this up here just for simulation purposes. When you're looking at traditional drug development, historically, randomized control trials have gone 1-to-1, 50% chance of being on treatment or placebo. And if you assume that 10% of therapies tested are effective with a 30% slowing of rate of progression in ALS, as measured by the ALS-FRS functional rating scale, you would need about 12 years, 10 treatments, and 2,400 participants with 1,200 on placebo. Whereas if you try to do the adaptive platform trial, which is what I showed previously, hopefully you could do 10 treatments tested over 4 years. And the big picture here is that you use less number of participants, 1,600, but even far fewer people on placebo. And I think that's the essence of the platform trial. But I didn't want to present everything there. That could be its own talk in and of itself. And then I just wanted to highlight this. I actually am not going to talk about tofersen because there's so much going on. I'll just talk about it just in the context of phases of clinical trials. But both tofersen, which is an antisense oligonucleotide for SOD1 ALS that's in clinical trials, and AMX0035 were both published in the New England Journal of Medicine. And most of you are familiar with sodium phenylbutyrate and terso, but essentially these were combined into one formulation to be tried in a clinical trial, phase 2, that was carried out with the PI being Sabrina Paganoni. She presented the data. And so this is just a quick snapshot. The full data is obviously in the New England Journal of Medicine and also on multiple webinars that you can easily access publicly. But the primary endpoint was looking at the slope of decline between placebo and the treated arm over 6 months. And so the difference was 0.42, but it was statistically, the p-value was 0.03. Now, if you look at that, that was the slope, but if you look at it by change from baseline, the change equated to about 2.32 difference. And just to put that into context, the edaravone study, the difference over 6 months, placebo versus edaravone treated, was 2.49. So it's in the similar realm of change from difference between placebo and treated arms. And the safety profile was pretty favorable. In fact, there was fewer serious adverse events in the treated arm versus the placebo. But the study data is available, but why I put it here is not necessarily to talk about the trial or the compound itself, but it begs the question that this study hits its pre-specified primary endpoint, and it was considered a phase 2. It's small in terms of study size compared to phase 3 clinical trials. But the pre-specified primary endpoint and pre-specified secondary endpoints fall in the correct direction, favoring treatment with a very good safety profile. And so why I put it here is that it starts to ask the question, how much evidence is enough evidence to consider access to patients or approval, or how much evidence is not enough evidence to ask the question, should we do another clinical trial? And in this case, when we have pre-specified endpoints that all look like they help slow down disease progression that's orally administered as opposed to intravenous or intrathecal with a very good safety profile, it would be difficult to conduct another clinical trial in the US when people feel that people living with the disease have a sense of a great unmet need. And so it becomes a difficult question to do another phase 3 clinical trial, especially when one of the components of the drug is easily obtainable over the counter, especially in the US. So it starts to push the thinking about what we would think is substantial evidence versus not substantial evidence for either full approval or provisional approval.
But
Go ahead.
doesn't that go against the fact that 80% of phase 2 trials fail when tried again, regardless of how good this might look? I mean, this
True.
is not a the equivalent of an SMA. This is still a relatively small effect.
Right. And I think a lot of the question goes back to like the compounds themselves and how, what is the rationale of putting them together? There isn't a lot of information about that. And so we know that the targets that they're aiming for are things that we know are relevant for ALS. But you're right. It asks the question, do we do it? And is it feasible to do it? You know?
Let me ask a follow-up. I'll ask you to put your pharma hat on. I'm not asking you to speak for Amylyx necessarily, but I guess one question might be, well, let's say it gets approved. Would a company be concerned that it might be great for the first year, but then it might tend to lose legs? In other words, people might say, well, it really doesn't work that well. I didn't see a difference. And so wouldn't a longer trial give you more evidence, I guess, if you have confidence in your drug? How do you think about that?
I'm glad you posed that question, because the reality is that that's a real risk, right? So say you get full approval or provisional approval of this, and say after a year or two, people feel that it doesn't seem to be helping them, you get less people on the medication, so you have less penetration in the market, so then you end up losing money on a drug that you thought would help patients living with ALS. And then it's happened actually with another example, Bevixa. It is a drug that was approved by Portola. They did an adaptive trial design, and their adaptive design didn't show effect, but when FDA reanalyzed it in 2018, they gave an unexpected approval. And so they went to a crowded market, and they were not able to penetrate the market as much as they hoped it could. And eventually, because of that lack of penetration into the market, they had to withdraw the product. And that's a risk if that happens in ALS, if you do something that is modestly effective and doesn't actually penetrate, because we have Riluzole, we have Edaravone already there. The other way of looking at it from a pharmaceutical perspective is, if we want to do another trial to reduce that risk of not penetrating the market, we do another trial. How is it feasible to do it in the US? Is it going to be difficult to recruit? So if we want to replicate this study, it might be that we would do the same design over, but it might take twice as long, and that costs a lot of money too. Right. So for smaller companies, it might be difficult to do that. Right. For larger companies, they can do that. Then if you're a pharmaceutical sponsor, you got to think a little bit out of the box. If you're a small company and you think feasibility might be difficult and the cost might be too high to run a very lengthy study where it'll be slow to recruit, you could say, I'm not speaking for Amylyx, I'm just speaking just as from my experience. One could say that you could since this was a US based study in US patients, and US patients are very excited about it, and it might be not feasible to enroll here, maybe we can do another phase 3 confirmatory study outside the US, in Canada or Europe or both, and then try to seek provisional approval or do a compassionate use program here in the US in the meantime, while you're collecting more confirmatory data, so that you don't unnecessarily, or you might not approve a drug that may not actually be effective. I mean, that's one way to think about it. So these are the things that people think about when they're trying to figure out how to get to market. Does that make sense?
Yeah, absolutely. It's an interesting perspective.
So certainly, you're right. And Jeff's had a great point. It's true that when you repeat a phase 3, there's high likelihood of not confirming the signal that you saw in the previous study. But in the case of ALS, you have to think about not only the scientific question of efficacy, but how is that drug administered, and what's the risk of that route of administration? What is the adverse event profile and tolerability profile? And what is the feasibility of conducting that clinical trial? Those are other aspects of moving it forward to either a provisional approval or to another clinical trial from a pharmaceutical standpoint. And I think that's just probably one of many. The cost of manufacturing the drug, the distribution, all of that goes into play as well. But that's an excellent question. And then I just put these here as placeholders, but it just goes to show that the patient voice and the results of the tofersen early phase clinical trials, and also Amylyx, and other clinical trials coming around the corner. Orphazyme will be reporting out in the first quarter of 2021 with a drug called arimoclomol. Brainstorm just presented their data in the International ALS M&D meeting on December 9th, and it all kind of asks the question about how to handle this, and the patient voice has kind of created movement both on the regulatory sphere and also on the legislative sphere. So there are two bills that are moving forward, and one is the Promising Pathway Act, which is a provisional approval pathway, different from accelerated approval pathway, but it does require companies. So the fear sometimes with FDA is that if they give accelerated approval, the company won't complete all of its post-marketing requirements. And so the provisional approval through the Promising Pathway Act will really tie the companies to get that post-marketing data. But it has to show substantial evidence of safety and early evidence of effectiveness. And it is a provisional approval, it's not a full approval. But this pathway would be available for all drugs, not just for ALS drugs. And again, the provisional approval is to establish a time-limited provisional approval, not something that would just stay provisional forever. So trying to get manufacturers to be responsible about getting the data that's required to give us as clinicians and people living with the disease good comfort on the fact that they've done their due diligence to determine efficacy of that drug. And then the other is that there's been a push specifically on the ALS front on trying to increase access to investigational products. This is putting pressure on us as clinical researchers and basic scientists about how we develop targets and how we develop drugs in clinical trials and how we move to the market. And so this is a bill that's supposed to help increase access by providing funds to allow for sites to provide expanded access or compassionate use to investigational therapies. And so this would involve input from actually ALS experts and experts from the neurodegenerative fields of Alzheimer's disease and Parkinson's disease, but also the NIH and the FDA in terms of prioritizing. Cancer field has a center of excellence very similar to this. And so this way there can be an evaluation of any potential asks for the money for compassionate use or expanded access. Compassionate use is the term in Europe, expanded access is the term in the US. And this would also create a center of excellence for neurodegenerative diseases at the FDA to do these evaluations and to support the Division of Neurology. But all this shift and changes in the regulatory landscape, as more complex molecules and biologics are investigated, I think we have to think really hard and creatively about the regulatory approaches when you're developing an investigational product to a specific target. So I put tofersen up there. I didn't talk about it because that could be its own talk too. But tofersen is an antisense oligonucleotide. You're doing a phase 1 study, but nowadays when you're doing any gene-directed therapy phase 1, you're going to have to, or even stem cell, when you're doing your phase 1 study, you should traditionally you do phase 1, phase 2, phase 3, but at that phase 1 level, traditionally you only look at safety, pharmacokinetics, tolerability, and you're really looking for safety and the behavior of the drug. But now when you're testing these specific gene-specific therapies or stem cell therapies, you really got to incorporate efficacy endpoints because that might be your pivotal study. And we've kind of shifted from doing the traditional phase 1, phase 2, phase 3 to maybe an early phase clinical trial and adapting that trial, if you see early efficacy, into a pivotal trial. So all of that information you gain right in your first in human studies is going to be very valuable for your registration or application for approval. Always considering the patient perspective, it's always important in the clinical discussions, even the clinical trial design, trying to consider if the development of that drug meets a public health imperative. And for ALS, certainly anything is important. You've got to try to get a meaningful therapy out there for ALS. But when you're looking at expedited pathways and accelerated pathways, that does not mean less rigorous regulatory review. You still need to bring the data, and the data has to be reviewed by the FDA. But it would probably require, if you do try to take the approach of getting accelerated approval, or if the Promising Pathway Acts go, a provisional approval, you'll have to do a lot of effort after approval more time, more effort, and probably more expense. And so that would be taken into consideration from a pharmaceutical perspective when you're trying to develop something in ALS, and the feasibility of collecting that post-marketing data. And the strategy will be to consider what's right for the company. Your portfolio, your company has a portfolio. If they have neurological drugs in the neurological space, they'll likely be favorable towards ALS. But they'll also look at what's right for the molecule, what's right for the biologic, and what's right for public health, especially for ALS and meeting that unmet need. So with that, I think I'm up on time, sorry. I can answer any other questions.
Jinsy, that was